In the previous lesson, we learned about innate immunity, the importance of skin and the mucosa, and how they block invading pathogens. How macrophages, along with neutrophils, combat enemy microorganisms that have breached our first line of defense. Macrophages are armed with PRRs, receptors that recognize structural patterns, PAMPs, that distinguish foreign threats from self, the components of our cells in our body, and non-self. Finally, we found out that not all bacteria are bad, and that our microbiome actually partakes in fending off unwanted intruders. When innate immunity is overwhelmed, however, and unable to successfully repel intruders, adaptive immunity kicks in. Two major aspects of adaptive immunity make it particularly effective and very different from innate immunity. First, the adaptive immune system is exquisitely specific. Its cells, the lymphocytes, are armed with sophisticated receptors that are able to focus and discern the finest structural details that precisely discriminate between us, that is, self, and pathogens, non-self. This unique capability allows the lymphocytes of adaptive immunity to get rid of the enemy with minimal collateral damage. Second is the fact that once T and B cells have learned how to best knock out a particular threat, they deposit memory cells in the lymphocyte archive. These memory cells are called upon in the event a pathogen attempts to infect us a second time. In this case, the pathogen is met with an immediate and strong response, clearing the threat before it can take hold and cause disease. So basically, the unique strength of the adaptive immune system is its memory and the ability to recall T and B cells armed with receptors, sophisticated, fine-tuned precision sensors. The B cell receptor, the BCR, contains a membrane-bound antibody. Antibodies, also called immunoglobulins, are relatively large proteins made up of 4 subunits, 2 heavy chains and 2 light chains, arranged in a Y-like shape. The 2 tips, where the heavy and the light chains meet, form the binding sites that recognize the antigen. Consequently, a single antibody has 2 identical binding sites. The stalk of the Y, where the 2 heavy chains meet, forms an effector signal. And as we shall see, this too is very important. The binding sites of an antibody recognize very specific yet limited structures of an antigen. These sites are called epitopes. Now, metaphorically of course, imagine an antibody whose epitope corresponds to the very tips of our fingers. This antibody would clearly read the details of our fingerprints, and although the binding is to a limited surface only, the antibody would be able to easily discriminate people with precision. Now, getting back to the BCR, the B cell receptor. The B cell receptor corresponds to the specific antibody that this particular B cell will ultimately produce and circulate in our blood. Each B cell can produce thousands of copies of its single one kind of antibody. Collectively, however, the billions of different B cells of our bodies can produce a vast variety of antibodies that can recognize every possible epitope in nature. When a pathogen encounters a B cell that just happens to have a receptor that can specifically bind one of its antigens, the system goes wild. This precise binding event triggers a process of clonal expansion. The specific bound B cell divides over and over again, generating thousands of B cells, all members of the relevant B cell clones specific for the pathogen. These activated B cells develop to produce and secrete millions of their specific antibodies. These will ultimately bind to the pathogen and neutralize it. Now, it turns out that antibodies can come in a number of different flavors and sizes and are adapted to specific tasks. Remember, in the very first lesson, we emphasized that life is all about structure and function. Antibodies have different structures, each with its special function. The very first type of antibody to be produced in response to the initial encounter of a B cell with a pathogen is called immunoglobulin M, or simply IgM. IgM has a binding site that recognizes its epitope with modest affinity. In order to compensate for this lower activity, however, IgM appears in the bloodstream as a complex of 5 copies of the antibody linked together to form a pentamer. Thus, although each IgM binding site may only loosely bind its epitope, together the pentamer's 10 binding sites work pretty well. Once the B cell binds its target antigen, the enemy, it can begin a course of basic training and learn how to best subdue the pathogen. This can take a few weeks. Through a process of trial and error, the genes that code for the IgM undergo local mutations and variations that refine and improve the binding of that antibody for its epitope. This process optimizes the binding site to become a super binder, which snugly fits its target, latching onto its epitope with tenacity and doesn't let go. In addition, the antibody itself converts to a lean and mean immunoglobulin called IgG. IgG antibodies are more effective. They live longer in our bloodstream and are quicker to bind and knock out pathogens, as we shall see. Another type of immunoglobulin are the IgA antibodies, which are especially adapted to survey and patrol our mucosa. IgAs are thus often the first antibodies to encounter pathogens and play an important role in clearing viruses that infect our gut, our lungs, and the urogenital tract. Finally, IgE antibodies are special antibodies designed to knock out and kill parasites. So as you see, the first B cell response produces large and somewhat clumsy IgM antibodies, but after a few weeks of training and maturation, antibodies become sophisticated guided missiles that intercept viruses before they infect our cells. Is this, however, enough to take out the enemy?